Hello, my name is Bryan Rodriguez, and today I am presenting the purification of the manganese oxidizing protein MOPE-HP found in the marine bacterium Erythrobacter SD21. I would like to start by first acknowledging the support provided by Caustic Fullerton, DNSF, Caustic Fullerton ASI, and my mentor Dr. Hope Johnson and her lab members that made this project possible. Manganese is an abundant transition metal found in both terrestrial and aquatic environments, and manganese oxides, because of their high redox potential, are some of the stronger oxidants in the environment, and they can react with the reduced form of other elements in the environment, such as iron, copper, and others, thus affecting their fate and their respective biogeochemical processes. This ability of manganese oxides to react with other metals makes it a good candidate for water and soil contamination treatments because it influences the toxic metal availability in the environment depending on other variables such as temperature and pH levels. And when I talk about manganese oxidation, I talk about going from manganese 2 plus to manganese 3 and 4 plus. And these two are the most abundant species of manganese found on Earth. And the image here on the right shows the manganese cycle of oxidation states found in nature. And here we observe that at high pH levels, manganese oxidation is favorable, as opposed to lower pH levels where manganese oxidation is kinetically inhibited. Manganese can also be oxidized by bacteria with the help of multiple peroxidases, peroxidase cyclooxygenases, previously known as animal heme peroxidases, and photosystem 2. Some of the manganese oxidizing bacteria include Pseudomonas putida, which a gram stain is included here on the right, Bacillus species, and Erythrobacter SD21. This last one oxidizes manganese with the help of a protein called MOPE. MOPE is a peroxidase cyclooxygenase that oxidizes manganese. And in the lab, we conduct a heterologous expression amplification of the peroxidase domain in MOPE, called MOPE-HP, which is around 130 kilodaltons. And for MOPE-HP to oxidize manganese, it requires the presence of calcium, heme, NAD+, and PQQ, which actually serves as a stimulant for optimal protein activity. Yet, the mechanism of manganese oxidation by bacteria, especially by MOPE-HP, is still unclear. Therefore, the objective of the project, as I mentioned before, was to conduct a heterologous expression amplification of MOPE-HP so that we can obtain a pure and active sample for further studies to have a better understanding of the manganese oxidation mechanism implied by this protein. So how do we do it? We overexpress MOPE-HP with the induction of L-ribinose growing in E. coli with an OD of 0.5 at 600 nanometers in alveoli medium containing kanamycin and chloramphenicol after the inoculation of an overnight growth at 37 degrees Celsius under shaking conditions. We spin down the solutions and resuspend the pellets in inky buffer, which contains Hebes, sodium chloride, and imidazole. We keep the samples under cold conditions from here on, and we lyse the cells with French press, and we spin down the cell-free extras and conduct different purification protocols. The first, I run my samples through a gravity nickel affinity chromatography, and my elutions, I run it through a PD10 desalting column to remove the imidazole and conduct a buffer exchange. This new buffer contains Hebes, sodium chloride, and glycerol. And lastly, my elution is run through an ion exchange chromatography column by hand to see if the protein will bind, and we also included avidin as a molecular clotting agent to help maintain the activity of the protein. All the samples collected were tested for protein presence through an SDS page, and protein activity was tested through a colorimetric LBB assay containing all the elements required that were previously mentioned, and activity assays were conducted in triplicates for better accuracy of the results. Here we have 6 different lanes in our SDS page, And in lane 1, we have a protein ladder, which we want to focus in the second band from top to bottom, which is the 130 kilodalton mark. Around this same area in every lane, we see a band which corresponds to the presence of MOPE HP. In lane 2, we have the samples obtained from sulfur extract obtained from the French pressing. In lane 3, we have the sample obtained from nickel affinity chromatography. Lane 4, sample obtained from the PDT desalting column. Lane 5 and 6 are the samples obtained from an ion exchange chromatography without and with adenine present, respectively. And we can see that in lane 3, we have a more pure sample of MOPE-HP present compared to number 2. And lanes 3 and 4, they look about the same purity levels, which is what we expected because we didn't make any purification attempt. It was just a buffer exchange. And in lane 5, although it looks more impure compared to lanes 3 and 4, it is actually purer because this is a concentrated lane as opposed to lanes 2 through 4, which are diluted in buffer. Same for number 6. This is a concentrated image of the sample that we collected. So we can see that in lane 6 we ended up with a clean, solid MOPE-HP band in the presence of adenine. From the protein activity assay, we see high protein activity in our sulfur extract protein sample, and we also see a major drop in protein activity in our nickel affinity chromatography elution, but activity is double when we conducted the buffer exchange, and this is expected as we hypothesized that the removal of the imidazole would result in an increase in protein activity. And lastly, there was no activity observed in either of the anion exchange chromatography samples collected, even in the presence of adenine. In conclusion, manganese is an abundant transition metal found in our environment, and manganese oxides, which have a high redox potential, can affect the fate of other elements in the environment required for other biochemical processes. And it can also affect the metal toxic availability in the environment, and this makes manganese oxidation a good bioremediation technique to employ in water and or soil contaminated areas. And manganese oxidation mechanism by bacteria and the proteins is still unclear, especially the MOPE-HP mechanism. Therefore, our goal was to express and purify a active sample of MOPE-HP to conduct further experiments to have a better understanding of its mechanism. And as it was demonstrated today, we were able to obtain what appears to be a clean band of MOPE-HP through an ion exchange chromatography in the presence of adenine, but we also had a loss of protein activity in the process. Clearly, something went wrong in between the buffer exchange and the anion exchange chromatography that led to protein activity being lost. So, current studies are focusing on figuring out what went wrong and fixing this issue so that we can conduct further purification steps to separate MOPE-HP from adenine, and a successful purification of an active MOPE-HP would allow us to conduct future studies to have a better understanding as to how MOPE-HP is capable of oxidizing manganese. And lastly, I would like to recognize the help and support by my mentor Dr. Johnson and my mentees Alisa and Natalia, who were really helpful throughout this project. Thank you.